Good afternoon, class. This is our last lecture on muscles and muscle tissue. So essentially, this is our last lecture before exam 3. And remember, exam 3 is going to cover chapters 6, 7, 8, 9, and 10. So exam 3 is going to cover chapters 6, 7, 8, 9, and 10. All right. So essentially, the skeletal system, joints, and muscles is going to be on the exam, and there will be some identification of joints, bones and skeletal muscles, so that you will have to ID structures on the exam. And the exam is going to be online, and it's going to open, so you have a window to take the exam. The window is going to start, it's going to open this Monday, April the 13th, and then the window closes 5 o'clock Thursday, April the 16th. Okay, and I understand most people aren't going to take the exam, and I wouldn't take the exam Monday, right? You always want to give yourself more time to study, but please don't wait until the last minute, and then you run out of time taking the test, something like that. So just be aware of that, and prepare yourself accordingly. All right, so far as factors that affect muscle contraction, so this is kind of intuitive. So obviously the numbers of muscle fibers stimulated. All right, so the more motor units recruited, the greater the force, and also the relative size of the fibers. The bulkier the muscle, the more tension it can develop. Okay, so that just follows physics 101. Okay, so and muscle size can increase in size with regular exercise. So lifting weights and things of that nature can increase the size of your muscles because you are demanding more from your muscles. Therefore, they grow in response to that demand that you are asking of your muscles. I also frequency of stimulation. So the higher the frequency, the greater the force, and the degree of muscle stretch. So muscle fibers with sarcomeres that are 80 through 120% their normal resting length generate more force. Okay, so if the sarcomere is less than 80% resting length, filaments overlap too much and the force decreases. If sarcomere is greater than 120% of resting length, filaments do not overlap enough, so force decreases. Okay, so the magic area is 80 to 120%. Less than 80 is bad. You're going to generate less force, and greater than 120% is also bad. You're going to generate less force. So this figure is just showing you a picture of what we got through talking about from the previous two slides. So if you recruit a large number of muscle fibers, okay, that's going to increase the force of muscle contraction. So all these things are going to increase the force of muscle contraction. If you have large size as far as muscle fibers, the increase, the larger the muscle, the greater the force of contraction it can generate. If you have a high frequency of stimulation, okay, that can create a greater contractile force. And then the degree of muscle stretch. So 80 to 120% is what you want. Around 80 through 120% is your degree of muscle stretch, and that's going to generate the greatest contractile force. All right, so how fast a muscle contracts and how long it can stay contracted is influenced by the muscle fiber type, the load and recruitment, and we're going to talk about muscle fiber types. Okay, and we're also going to talk about how load and recruitment play a part, but the muscle fiber type is very important for as far as there are certain fibers that are for endurance and certain fiber types that are more better suited to a short burst of energy. So now let's talk about the different types of muscle fibers. So muscle fibers are classified according to two characteristics. So the speed of contraction, whether it's slow or fast, fibers whether they contract slow or fast. Okay, speed of contraction. So and this is dealing with the speed at which the myosin ATPase split ATP. Okay, so again, the speed at which myosin ATPase splits ATP. So remember, if you go back into where when a sarcomere is contracting, okay, remember the myosin head is bound, it's coupled to the actin, okay? And essentially it stays, and then, it's going to make the actin filament slide, but it stays attached to the actin filament until ATP binds back to the myosin head, which causes decoupling. So it's going to cause when ATP binds, the myosin head now it's going to release the actin, okay? And then when that ATP gets split, okay, then that myosin head is ready to attach to another actin filament. Okay, so the speed at which myosin ATPase split ATP, okay, it's going to determine the speed of contraction, slow or fast fibers. Fibers that the myosin ATPase split ATP slowly are going to be slow fibers. Fibers that the myosin ATPase split ATP quickly are going to be fast fibers. And again, until that myosin ATPase splits ATP, that myosin head is not going to be ready to attach to another actin filament, okay, actin binding site, actin in a thin filament. All right, and then the pattern of electrical activity of motor neurons. All right, so then another way that you classify muscle fibers is their metabolic pathways, okay, how they use and how they generate ATP, okay. So the name tells you essentially how that occurs. Oxidative, all right. Think of oxygen. Oxidative fibers use aerobic pathways. Aerobic means oxygen pathways, and glycolytic fibers. Okay, glycolytic. Think about the glycolysis. That is, remember when you first break down the sugar. So when the sugar first enters the cell, all right, it's gonna get broken down to two pyruvate molecules through the glycolytic cycle, through anaerobic glycolysis, and it's called anaerobic glycolysis because essentially glycolysis doesn't need oxygen for this to happen. So again, when a sugar first enters a cell in the cytoplasm, it's going to go through glycolysis. And it's going to break the sugar down to two pyruvate molecules, and in this process, it will generate two ATP. Okay, and that first step of far as breaking down the sugar, anaerobic glycolysis, does not require oxygen. But and then we already talked about this, but I'll bring it up again. But if you wanted to continue to break down that pyruvate, all right, you can continue to break that down. It can go to the mitochondria, okay, and go through the TCA cycle, and then go through the electron transport chain. But once it goes to the mitochondria, it needs oxygen, okay. So that's aerobic respiration. It needs oxygen because the electron transport chain uses oxygen as the final electron acceptor, all right, generating water. So you can't go to the mitochondria. The pyruvate cannot go to the mitochondria without oxygen. Okay, so and we talked about that before, but I just want you to understand, anaerobic glycolysis is just that first step when you are breaking down sugar into two pyruvate molecules, and that happens in the cytoplasm. Okay, and that does not require oxygen, and it only generates two ATP. It doesn't generate a lot of energy either. It only generates two ATP. Okay, whereas aerobic respiration. Once that pyruvate goes to the mitochondria, goes through the TCA cycle and electron transport chain, it's going to generate roughly 32 ATPs. So aerobic pathways generate way more energy. Okay. All right, so based on these two criteria, skeletal muscle fibers can be classified into three types: slow oxidative fibers, fast oxidative fibers, or fast glycolytic fibers. And again, without even talking about more about this, we know we're dealing with slow and fast, which, remember, slow and fast tells you about, let me go back to the previous slide. Slow and fast tells you about the speed at which the myosin ATPase split ATP. So that's what the slow and fast tells you. And then oxidative or glycolytic. So oxidative, it's going to use oxygen. Okay. Glycolytic, it's going to be anaerobic. Okay. So without even going into more, we already can break that down into that. Okay. And most muscles contain a mixture of fiber types, resulting in a range of contractile speed and fatigue resistance. All fibers in one motor unit are the same type, and genetics dictate individuals' percentage of each. And just be aware that different muscle types are better suited for different jobs. Okay, so for instance, different muscle types are better suited for different jobs. Your slow oxidative fibers, essentially, they contract slowly because it's myosin ATPase activity is slow. Okay, it's oxidative because it depends on oxygen delivery for its aerobic pathways. It's how it generates ATP through aerobic pathways. So it's oxidative. It's these fiber types resist fatigue and has a high endurance capacity. Okay, they're thin. All right, it's they these fiber types has relatively little power, has many mitochondria. They have a rich capillary supply, and they are red in color. And they're red in color because they have an abundant supply of myoglobin. And myoglobin is the muscle's oxygen binding pigment. Okay, so they're red. So again, slow oxidative fibers. The color is red because they have abundant supply of myoglobin. They generate ATP through aerobic respiration. They contract slowly because of its myosin ATPase activity. They resist fatigue and have high endurance. They're thin and they generate relatively little power, and they have many mitochondria. An example of these fiber types will be used for maintaining posture. So this figure is just kind of summarize what we got through talking about. So your fast glycolytic fibers, okay, so they contract fast, but they are anaerobic, all right, so they fatigue quickly, okay, and then you have your slow oxidative, so they contract slowly and they depend on oxygen for ATP, all right, so they are resistant to fatigue, okay. And then the load plays a part on the velocity and duration of muscle contraction. All right. So this just, if you increase a higher contractile velocity, it's going to be your fast. They're going to contract quickly. Glycolytic fibers, anything with the word fast in front of it, and then your slow oxidative fibers are going to contract slowly, but they are going to be more built for duration, endurance. Okay, so they're going to be less resistant to fatigue. So as you would expect, all right. So far as loading recruitment, so you would expect muscles contract faster when no load is added. Okay. The greater the load, the shorter the duration of contraction, and the greater the load, the slower the contraction. And then far as recruitment, the more motor units contracting, the faster and more prolonged the contraction. So this is just a figure showing what we got through talked about in the last slide. So the greater the load, the briefer the duration of muscle shortening. So you can see right here, you have a light load, and you see how long the duration of shortening is, all the way up to 80 milliseconds. Seconds. Heavy load, all right, you're only going from 20 to 40, so only about 20 milliseconds. So the greater the load, the briefer the duration of muscle shortening, and then also the greater the load, the slower the muscle shortening. So you see right here at a very heavy load, you see that its velocity of shortening is decreasing. The heavier the load, the more it's going down. Okay, so the greater the load, the slower the muscle shortening. All right, so your muscles can adapt, right? You can train your muscles. You can train them for whatever sport you play, right? If you are a marathon runner, you can train your muscles for endurance. Okay, if you are a power lifter, okay, you can train your muscles for powerlifting. Okay, so your muscles do adapt to exercise. Okay, so um, aerobic exercise such as jogging, swimming, biking leads to increased muscle capillaries, number of mitochondria, and myoglobin synthesis. So they resist, and the result is greater endurance, strength, and resistance to fatigue. And you can convert fast glycolytic fibers into fast oxidative fibers. So remember, so they both start off with fast, so that's just the how fast they contract. But glycolytic fibers, remember, are anaerobic. They don't require they depend on glycogen reserves. They don't require oxygen for ATP generations, whereas the oxidative fibers rely on oxygen for ATP generation. Okay. All right, so resistance exercises, typically anaerobic, such as lifting weights, um, or isometric exercise, lead to muscle hypertrophy. Um, so that's due primarily to an increase in fiber size, increased mitochondria, myofilaments, glycogen stores, and connective tissue. Increased muscle strength and size. All right, so those are your resistance exercises. And again, they're typically anaerobic. All right, so just a clinical note. So muscles must be active to remain healthy. So if you don't use your muscles for whatever reason, you got into some kind of accident and you couldn't um, walk for several months, for instance, all right, you can expect some kind of atrophy. So loss of muscle mass. Um, so you can also be due to some type of neurological diseases. Also, your muscles have to get stimulated, right? By your skeletal muscles get stimulated by neurons. So if you have some kind of neurological disease, also that can lead to muscle atrophy too. Okay, um, so muscle strength can decline 5% per day. So paralyzed muscles may atrophy to 1/4 initial size. Fibers, connective tissue replaces lost muscle tissue. and rehabilitation is impossible at this point. So this is just showing a summary of skeletal, cardiac, and smooth muscle. And again, when we talked about at the beginning of the muscle tissue lectures, we talked about briefly differences between skeletal, cardiac, and smooth muscle, even though we spent the majority of our time talking about skeletal muscle. But again, you should know the difference. Skeletal muscle has striations, cardiac muscle has striations. Um, but cardiac muscles have those intercalated discs, so you can tell them apart. Smooth muscle has no striations. Cardiac muscle is only found in your heart, and smooth muscle is found in your autonomic visceral organs. Your smooth muscles, digestive tract, bladder, things of that nature. All right. And it table talks about other differences between um, skeletal, cardiac, and smooth muscle. So pretty much the presence of myofibrils composed of sarcomeres. So remember, in your skeletal muscle cell, um, they call muscle cells fibers because they're long. Okay, but your skeletal muscle cell, you have an organelle, the myofibrils. Okay, so the skeletal muscle cells are packed with myofibrils. And cardiac has myofibrils, but they're irregular thickness. And smooth muscle does not have myofibrils, but actin, myosin filaments are present throughout. Dense bodies anchor the actin filaments. All right, and then presence of T tubules. So skeletal, yes. Cardiac, yes. Smooth, no. All right, so some more comparisons of the three. So, does it have a elaborate sarcoplasmic reticulum? Skeletal, yes. Cardio, less than skeletal muscle, and then smooth, equivalent to cardiac muscle. All right, so it does have some SR, um, sarcoplasmic reticulum. Presence of gap junction. Skeletal, no. Cardiac, yes, at the intercalated disc. And smooth, yes. Cells exhibit individual neuromuscular junction. Skeletal, yes. Cardiac, no. Smooth, not in unitary muscles, yes, in multi-unit muscle. And then regulation of contraction. Skeletal, voluntary via axon terminals of the somatic nervous system. Cardiac, involuntary, and smooth, involuntary. All right, so um, some more comparisons. So source of calcium for calcium pulse. Skeletal calcium gets released from the sarcoplasmic reticulum. Cardiac from the sarcoplasmic reticulum and from extracellular fluid. And smooth muscle from the sarcoplasmic reticulum and from extracellular fluid. Site of calcium regulation. Troponin on actin containing thin filaments for skeletal. Troponin on actin containing thin filaments for cardiac, and calmodulin and inositol for smooth muscle. And again, I know we haven't talked a lot about smooth muscle, so I can go ahead and tell you, you're not going to be tested on far as knowing that aspect of smooth muscle. You just need to understand that smooth muscle is involuntary, no striations, and you find it in hollow digestive organs and also your bladder and things of that nature. Okay, but when we talked about things that we did talk about, and like I said, we talked mostly about skeletal muscle. You should know that for skeletal muscle. Um, Presence of pacemaker, skeletal no, cardiac yes, smooth yes. Effects of nervous system stimulation, excitation, excitation or inhibition for cardiac, and excitation or inhibition for smooth. All right, speed of contraction for skeletal, slow to fast. For cardiac, slow. For smooth, very slow. All right. All right, so just the developmental aspect of muscles. So all muscle tissue develop from embryonic myoblasts. Multinucleated skeletal muscle cells formed by fusion of many myoblasts. Growth factor stimulates clusters of acetylcholine receptors at neuromuscular junctions. Cardiac and smooth muscles myoblasts do not fuse but develop gap junctions. Cardiac muscles start pumping when embryo is 3 weeks old. So this is showing you the myoblast fuse to form a multinucleated skeletal muscle fiber. All right, so embryonic mesoderms. So first embryonic mesoderm cells called myoblasts undergo cell division. Okay, so now you have your myoblast. Then several myoblasts are going to fuse to form a myotube. Okay, so you got the myotube. And then the myotube is going to mature into skeletal muscle. All right. All right, so regeneration of muscle, myoblasts like skeletal muscle, satellite cells have limited regenerative ability. Cardiomyocytes can divide at modest rate, but injured heart muscle is mostly replaced by connective tissue. That's why it's bad when you have a some kind of heart attack. Um, when you damage your heart muscle, it's usually replaced by connective tissue, which isn't good. And then smooth muscle regenerates throughout life. Cardiac and skeletal muscle can lengthen and thicken in growing child and adults. That can lead to hypertrophy. All right. So if your heart is working harder because your arteries are clogged and your heart has to pump harder because your arteries are clogged to move your blood, okay, that can result in hypertrophy of your heart. And hypertrophy heart, if it continues, it can result in heart failure. Okay, so that's not a good thing. Muscular development in infants reflects neuromuscular coordination. Development occurs head to toe and proximal to distal. So a baby can lift its head before it is able to walk. Peak nature, peak natural neural control occurs by mid-adolescence. Athletics and training can continue to improve neuromuscular control. All right, so there are differences in muscle mass between sexes. So female skeletal muscle makes up about 36% of their body mass, and male skeletal muscle makes up about 42% of body mass, primarily as a result of testosterone. So males have a greater ability to enlarge muscle fibers, also because of testosterone, and body strength per unit muscle mass is the same in both sexes. So with aging, so with age, um, connective tissue increases and muscle fibers decrease. So by age 30, you do start having loss of muscle mass, but no, don't fear the regular exercise. You can reverse um, some loss of your muscle mass. And atherosclerosis is how when your arteries get blocked, may block distal arteries, leading to intermittent claudications, limping, and severe pain in leg muscles. So that's essentially atherosclerosis can clog your arteries. So when your arteries get um, um, fatty, the fatty layer of your arteries hardens, okay, and it clogs, essentially makes your arteries clogged, more narrow, and that's going to lead to um, a less um, blood flow. Your blood flow is going to be decreased, okay, and so your distal um, body parts like your legs, and so far may um, have insufficient blood flow, and you can end up with pain in your leg muscles and so forth. All right, if you have bad blood flow because your arteries are clogged, essentially. All right, muscular dystrophy is a group of inherited muscle destroying diseases. Generally appear in childhood. Muscles enlarge as a result of fat and connective tissue deposits, but then atrophy and degenerate. Duchenne muscular dystrophy is the most common and severe type, caused by a defective gene for dystrophin. It's inherited through sex-linked trait carried by females and expressed by males. So dystrophin is a cytoplasmic protein that links the cytoskeleton to the extracellular matrix, stabilizing the sarcolemma. Remember, sarcolemma is the plasma membrane of the muscle fiber. So a fragile sarcolemma tears during contraction, causing entry of excess calcium, which leads to damaged contractile fibers. Inflammatory cells accumulate and the muscle mass declines. Victims become clumsy and fall frequently, and it's going to usually appear between ages 2 and 7. So currently, there's no cure. Prednisone can improve muscle strength and function. Myoblast transfer therapy has been disappointing. Coaxing dystrophic muscles to produce more utrophin has been successful in mice, and viral gene therapy and infusion of stem cells with correct dystrophin genes show promise. Patients usually die of respiratory failure in their early 20s. All right, so now we're gonna start talking about different types of muscles. So you have prime mover, so muscle with the major responsibility for a certain movement. You have an antagonist, muscle that opposes or reverses a prime mover. You have synergist, muscle that aid a prime mover in movement and helps prevent rotation. And you have fixtor, stabilizes the origin of a prime mover. All right, so this is an example of. So this figure showing you an example of a muscle that crosses. So muscles that cross um, on the interior side of a joint produces flexon. So for instance, this pectoralis major, um, you see this anterior view is producing flexion. A muscle that crosses on the posterior side of a joint produces extension. All right, so this is the posterior side of a joint, okay? Um, and you see the latissimus dorsi, is going to produce extension, okay? All right, a muscle that crosses on the lateral side of a joint produces abduction. Example, the medial deltoid muscle, okay? Abduction, all right? It's moving the arm, moving the shoulder up, okay, and away from the body, moving it away from the body. And a muscle that crosses on the medial side of a joint produces adduction. An example is teres major muscle, okay, and that's going to that's a muscle that crosses on the medial side of a joint. It's going to produce adduction. All right, so the arms are moving toward the body. All right, so how they name skeletal muscles. So they name them by different ways, but their name gives you clue to what they do. So they can name them by the direction of muscle fibers. For example, rectus is straight, by the relative size of the muscle, maximus is largest. Okay. By the location of the muscle, temporalis, the temporal bone. By the number of origins, triceps has 3 heads. Okay. All right. By location of the muscle's origin and insertion, sternal, for example, on the sternum. By the shape of the muscle, deltoid, triangular, and by action of the muscle, flexor and extensor. By flex or extends a bone. All right, so this is just a diagram showing you um, the names of different muscles. All right, and it's showing you, giving you a description, okay, of certain origins and how the fascicles um, function essentially. So fascicle arrangement determine the muscle shape and force. So all skeletal muscles consist of fascicles, and if you remember from our previous lecture, um, a fascicle is just a bundle of muscle fibers, okay? But the fascicle arrangement vary, okay, resulting in muscles with different shapes and functional capabilities. And the most common patterns of fascicle arrangements are circular, convergent, parallel, and pinnate. Pinnate. And we're going to talk about each of these. I'm going to briefly talk about each of these fascicle arrangements. So, with the circular fascicular pattern, all right, so the fascicles are arranged in concentric rings, and muscles with this arrangement usually surround external body openings, which they close by contracting. So, examples would be the example of the circular fascicular arrangement, or the orbicularis muscles that surround the eye and the mouth, the eyes and the mouth. Okay, you find these orbicularis muscles. All right, so now let's talk about fascicles that are convergent muscles. Okay, so a convergent muscle has a broad origin, and its fascicles converge toward a single tendon of insertion. Such a muscle is triangular or fan shaped, like the pectoralis major muscle. All right, you see how it's fan shaped, pectoralis major muscle, and you see how that the muscle is broad, has a broad origin, and the fascicles are converging. You see how they're converging toward a single tendon of insertion. All right, so fascicles that are arranged in a parallel arrangement, the length of fascicles run parallel to the long axis of the muscle. Such muscles are either strap-like, the sartorius muscle of the thigh, or spindle-shaped with an expanded belly, like the biceps brachii muscle of the arm. Some anatomists actually classify the spindle-shaped muscles into. You see, this is spindle-shaped muscle right there, and then below it, you see the strap-like muscle, the parallel long strap-like muscle. So essentially, some anatomists actually classify the spindle-shaped muscles into a separate class. They call them fusiform muscles. All right. So just be aware of that. And on this figure, they actually have the biceps brachii, spinal-shaped muscle, classified as a fusiform. Okay. And then they have the long fascicles parallel to the long axis. They have the sartorius as an example of a parallel muscle type. So they have them classified separately. All right. And you also see how your spinal shaped muscles are tapered on both ends with an expanded belly. Okay. Um, but the fibers, the fascicles, are running parallel. Okay. But because they're spindle shaped, um, they call them fusiform. So means spindle, spindle. So essentially, they just call them fusiform because of that. All right, but they are parallel. They are parallel fascicles, but because of their shape, they call them fusiform. Okay. And the last fascicle bundle arrangement is pinnate. So in a pinnate pattern, the fascicles are short and they attach obliquely. You see how these muscle fibers attach kind of at a incline. You see how they're not straight. They're not straight. They don't attach straightly, and they're not perpendicular. See how they're inclined. Okay, that's obliquely. They attach obliquely to a central tendon that runs the length of the muscle. And pinnate muscles come in three forms: unipinnate, bipinnate, and multipinnate. All right, so the unipinnate fascicles insert into only one side of the tendon. Um, so an example of that is the extensor digitorum longus muscle of the leg. Okay, so you see the fascicles are short and insert into only one side of the tendon. Bipinnate, right here, bipinnate is the shape in which the fascicles insert into the tendon from opposite sides, so the muscle looks like a feather. All right, so you see right here the fascicles are short and insert into the tendon at opposite sides, so it looks like a feather-like. All right, so the rectus femoris of the thigh is a bipinnate example. Rectus femoris, okay, a thigh muscle. The multipinnate right here. All right, so multipinnate, which looks like many feathers side by side, with all their quills inserted into one large tendon. All right, the deltoid muscle, which forms the roundness of the shoulder, is a multipinnate muscle. All right, so again, fascicles are short and insert into one large tendon, feather-like deltoid muscle, an example. All right, so now we're going to talk about some specific muscles, and remember, anything in red is important. Frontalis is a facial muscle, raises the eyebrows. Orbicularis oculi, circular muscle, closes the eyes, blinks, winks. Orbicularis oris, that's going to close the mouth, is another circular muscle. Zygomatic muscle raises the corners of the mouth. Okay. All right. And then the buccinator muscle flattens the cheeks and chews. Okay. Other chewing muscles, masseter, closes the jaw and elevates the mandible. Temporalis, synergist of the masseter, closes the jaw. Neck muscles, platysma pulls the corners of the mouth inferiorly, and the sternocleidomastoid mastoid flexes the neck, rotates the head. All right, and so this shows you where the muscles are located. Now, hopefully, because we've discussed muscles in lab, and if you have lab with me, I know some of you have lecture with me, but you don't have lab with me. If you have lab with me, I told you when we went over these labs to go ahead and start memorizing these muscles. Okay, so essentially, the names, like we told you certain ways that you can name, like frontalis. Okay, it's in the front. Orbicularis, you know it's a circular muscle. Or so the names can help you. Temporalis in the temporal region of the skull. Sternocleidomastoid mastoid muscle. So the names can help you. All right, masseter, orbicularis oris, buccinator. All right, so the names do help you. Platysma. All right, but there still is a lot of being able to visualize it. You have to be able to visualize it on, in this case, a model. So if you started and you went ahead and been reviewing your muscles, like I hope you have, because I gave you a muscle list that you need to know for lab, and these are a lot of the same muscles that you need to know for the lecture, okay? Because there are on the test, you will have to identify certain muscles, and they're possible that you have to identify certain joints and bones too, okay? So what I'm trying to get to is if you haven't started reviewing, okay, you're gonna be already kind of behind, because is really no shortcut to learning these muscles in their locations. You just have to study, and I um, always advocate using flashcards, and that flashcards that you make up yourself. I'm talking about flashcards, those little gray anatomy flashcards, or flashcards that you can get on your phone, and you can use your phone to flip through it and study it that way. Okay, that's a good way to study it because you will have to ID muscles, bones, and joints on the test. Okay. All right. So muscles of the trunk, shoulder, and arm. Anterior muscles: pectoralis major, adducts and flexors the humerus, and intercostal muscles. You have external intercostals, raise the rib cage during inhalation when you inhale, and the internal intercostal, which depress the rib cage to move air out of the lungs when you exhale. All right, so this is just showing you the location of the pectoralis major muscle. All right, so muscles of the abdominal girdle. So the rectus abdominis flexes a vertebral column and compresses abdominal contents. The external oblique flex the vertebral column and it rotates the trunk and bend it laterally. The internal oblique flex the vertebral column and rotates the trunk and bend it laterally. And transversus abdominis compresses the abdominal contents. All right, so this shows you the muscles, the rectus abdominis right here, transverse abdominis. You see how that's going transversely. The internal obliques, you see that's, you remember oblique, they run, they're inclined, okay? They run in an incline. You see how that's in an incline? Internal obliques and then external obliques, okay? You see how they're inclined. All right, posterior muscles, trapezius elevates the press, adducts and stabilizes the scapula. Latissimus dorsi extends and adducts the humerus. Erector spina, back extension. Quadratus lumborum flexes the spinal laterally, and your deltoid, arm abduction. Again, the ones in red are the ones that are important. Remember, things in red. All right, muscles that arise from the shoulder girdle and cross the shoulder joint to insert into the humerus include pectoralis major, latissimus dorsi, and the deltoid muscles. All right, so this is your trapezius, all right, muscle, deltoid muscle, latissimus dorsi, all right, muscle, all right. All right, so you have erector spinae muscles, iliocostalis right here, iliocostalis, all right, longissimus right here, and spinalis right here. Okay, spinalis you see attached to the spine of the vertebrae. Iliocostalis, you see that's close to the costal cartilage, and longissimus, you see how it's the muscle is essentially long, all right, and quadratus lumborum right here. All right, muscles of the upper limb, biceps, brachii, supinates the forearm, flexes the elbow, brachialis, elbow flexon, brachioradialis, weak muscle, elbow flexon, triceps brachii, elbow extension. It's an antagonist to the biceps brachii. So this is showing you on the figure. So the biceps brachii. Remember that's the fusiform muscle. Brachialis. You see muscles right there. Brachioradialis muscle right here. Triceps brachii right there. All right, so muscles of the forearm which insert on the hand bones and cause their movement include the flexor carpi, which flexes the wrist, flexor digitorum, finger flexion, extensor carpi, wrist extension, and extensor digitorum, finger extension. All right, muscles causing movement of the at the hip joint include the gluteus maximus, hip extension, the gluteus medius, hip abduction, iliopsoas, the hip flexing keeps the upper body from falling backward when standing erect, and the adductor muscles which adduct the thighs. All right, gluteus medius, gluteus maximus, adductor magnus. All right, showing you those muscles, and this is the iliotibial tract. So in safe area to inject is the gluteus medius. All right. You see running back here through the gluteus maximus. You have the sciatic nerve, and anybody who's ever um, had back pain, a lot of times it stems from that sciatic nerve. Um, all right, so the iliopsoas, you have the psoas major right here, iliacus right here, the adductor group right here. And this also shows you the quadricep muscles. So the quadriceps is a group of four muscles in the front thigh. All right, you have the rectus femoris right here, vastus lateralis right here, vastus medius right here, and there's also a vastus intermedius that you can't see, which is going to be underneath essentially the rectus formalis, but you can't see it. But that makes up your quadriceps. All right, so muscle of the lower limb, you have muscles that cause a movement at the knee joint. You have your hamstring group, which causes thigh extension and knee flexion. Your biceps femoris, semimembranous, and semitendinous muscles. All right, so here go your hamstring group, biceps, for femoris right here, semitendinous right here, and then semimembranous right here. All right, those make up your hamstrings. All right, so muscles cause movement at the knee joint. Sartorius flexes the thigh, and then the quadriceps groups extends the knee. Rectus femoris. Then you have three vastus muscles: vastus lateralis, vastus medialis, and vastus intermedius. And we already talked about that. Sartorius muscle right here. Um, then your quadriceps rectus femoris right here, vastus lateralis right here, vastus medius right here, and there's a vastus intermedius that runs underneath the rectus femoris that you can't see. Okay, you can't see the vastus intermedius. The vastus lateralis is another good site for injections. All right. All right, so muscles cause movement at ankle and foot, tibialis anterior, dorsiflexon, foot inversion, extensor digitorum longus, toe extension, and dorsiflexion of the foot, fibularis, muscles, plantar flexion, foot eversion, soleus, plantar flexion, gastrocnemius, the plantar flexon. All right, so this is showing you the tibia bone, soleus muscle, fibularis longus muscle, fibularis brevis, tibialis anterior muscle, extensor digitorum longus muscle, fibularis tertius right here. and it's showing you the gastrocnemius muscle, all right, and soleus muscle. Okay, and then survey of muscles you need to know. And again, um, this is the anterior view, and there's also a posterior view. All right, and it is a lot of muscles, but like I said, hopefully you have already been reviewing your muscles, um, because you had to review your muscles for your anatomy lab. So hopefully you've already been reviewing your muscles, and this is just a review, and you already know these muscles. But if you don't know them, then you need to know, be able to ID them before the test. Okay. But like I said, hopefully you've already been studying the location, the names and location of these muscles. So it'll just be a review for you. All right. So again, this is the last lecture before the test, which opens up Monday, April the 13th, and you have until Thursday, April the 16th to take it. I think it's 5 o'clock Thursday. So again, please have enough time, leave enough time for you to take the exam. And it is going to be an online exam, but it will be timed. Okay, so you're not going to have an infinite amount of time to take the test. So it is going to be timed, a timed exam, and you can also possibly expect some short answer too. It's going to be multiple choice. You're going to have to ID muscles and bones and joints, and um, and possible some short answer too. Okay, but it will be timed exams, so don't think that you have all the time in the world to answer a question. Okay, and also I know with the lab exam we are not allowing you to go back, so I'm not sure if we're going to do the same thing for the lecture exam. I would think probably we're probably going to do the same thing. So just expect that you probably will not be able to, after you answer a question, to return to that question. I know we were doing that for the lab exam, and we're probably going to do the same thing for the lecture. All right. Thank you.